talking about extractable and leachable strategies and how there is a synergistic role of the risk assessment to the analytical testing. So here, the agenda of my speech of today. So we will start with an overview on extractable, leachable, and the key principles of the risk-based approach, and how the risk matrix is fundamental and is a multifactorial pathway, and is fundamental to depict the correct extractable and leachable strategy, and the risk proportionate evaluation, of course, finally to the analytical approach. So, start talking about the extractable and leachable. So, who they are. Extractables are chemical compounds that can be released from a contact material or a primary packaging under aggressive or exaggerated laboratory conditions. So, it's a worst case condition. While for the leachables, they are chemical compounds that actually migrate into the drug product or to the patient during normal condition of use, within also, for example, medical devices or the device used by the patients. So, in an imaginary world, extractable and leachables are in the same frame. But in the real world, we see that the leachables and the extractables can depict a fingerprinting, but not of all leachables, because we have to consider that the drug product can have chemical interaction with the contact materials and the primary packaging. So we have some compounds that can be excluded by the extractable part. So it's quite important to have the whole imagine of the possible migration of these compounds and contamination. And which are the main actors? So the application of this kind of testing. We see that we have different actors. First of all, of course, the primary packaging and the secondary packaging. So all container closure system, the labels and the cartons. And of course, also drug delivery components like pumps, intravenous bags, inhalants, and so on. So the first two actors are quite important because they are directly in contact with the patient. So the concern is the highest concern for these two main topics. But we have also to consider the manufacturing components like the single-use systems and the multi-use systems, so tubings, filters, gasket, and so on. Every plastic components that can come in contact with the formulation during the manufacturing process. And of course, we must also consider the medical devices, so all catheters, implants, surgical instruments, and also liquid medical devices. So every part that come in contact with the patients are can be part of this kind of project. And identify and understanding this kind of contamination is crucial for the pharmaceutical, biopharmaceutical, and medical device development cycles because, of course, it can ensure the safety, the stability, and achieves, of course, in the first step, a regulatory compliance across all the components. And why extractable and leachables projects are crucial, so why they matter. We have different topics to talk about as issues. Of course, the product quality impact, because these kind of compounds can interact with the formulation and altering the potency or even compromising the device performance. So it's quite important to understand and identify them. And of course, we can have real-world consequences. So the worst case can be, for example, a recall of the drug product due to this kind of contamination. And these events, of course, carry a significant regulatory and financial repercussion. And moreover, we have stability concern, because this kind of chemical interactions can affect, for example, shelf life, or can reduce the efficacy and create harmful byproducts. And finally, but not least, the patient have risk because leachables may be identified as toxic or even worse, carcinogenic compounds. So, and even traces amounts can trigger serious, for example, allergic reaction. And for this reason, the patient health becomes the primary purpose of this kind of project. So we have to take in mind some key principles, of course, in a risk-based approach. And the first is, of course, to protect the patient's safety. So the evaluation of this kind of contamination has to be based on all potential toxicological impact. And we can do it considering a science-based decision-making. So using all toxicological tools and thresholds, both in the toxicological overview and, of course, also analytical overview. And we have to have a strong understanding on the product and the process too. So we have to build a strong knowledge about the formulation, the container closure system, all the contact materials that come in contact with the drug formulation during the manufacturing process, and all the factors that can impact on an eventual migration of these compounds. And a life cycle management, because risk evaluation has to be performed from the early development of the drug formulation, considering an early material selection, and the screening of the product qualification to the post-approval changes. So it's a dynamic evaluation and not a static one. And what about the regulatory landscape? You can see that we have different guidelines, each for a primary packaging or a single-use system for biopharmaceutical products, primary packaging. So we have a different framework, and how we can manage this non-homologous framework. Fortunately, this year, on August, a guideline by ICH for extractable and leachable was released. So we are waiting for this. We were waiting for this draft, the ICH Q3E, and this draft depicted the frame of the quality risk assessment, risk management process, starting from the ICH Q9. So we can see that the risk management is divided in three main paths. So the risk assessment as in the first part, a risk control with the risk reduction and the risk acceptance, and finally the life cycle management. But looking specifically at the risk assessment, we have three different topics to talk about. The first is the hazard identification. And within the hazard identification, we have to know all information about all the potential interaction with the formulation and all the pretreatments of the components that must be identified and take in mind. That we move to the risk analysis. So, which is the risk? We know that the risk is the hazard mixed to an exposure evaluation. So in this case, we have two main points to take in mind. So the first is the manufacturing and conditions and the storage condition too. And also the propensity of the formulation to leach into the manufacturing process materials or the primary packaging. And finally, the last part, the integrated risk evaluation. So after the hazard identification and the risk analysis, we have to know something about the chemical characterization, safety, and the quality aspect. And to do that, we have to balance the potential harm severity and the acceptable risk threshold. And to do that, we have three main points: the quality, the safety, that goes directly to the efficacy of the drug formulation. And these three main points, so the risk assessment, has two main paths that we have to use. The first tool is the prior knowledge. It's quite fundamental to have a prior knowledge of the manufacturing components, the process, and the drug formulation. So every information is crucial to build the correct and optimized extractable leachable project. And to do that in the best way, we have to close collaborate. So all the parties involved, from the supplier to the industry to the analytical part and the toxicological part, has to strictly collaborate. And to do that, of course, we have to manage a risk matrix that is multifactorial. You can see that we have different aspects to take in consideration, but in two main topics: the pharmaceutical quality consideration and the safety assessment consideration. So you can see that in this matrix we have different factors to take in mind. So from the process to the evaluation of every step of the process and the factors that can interact and impact on an eventual leaching of the contact materials, and also in a safety assessment all the characteristics, the main characteristic of the drug formulation. So the route of administration, the duration of the treatment, the leachable dose exposure, and so on. So how can we move from risk assessment to the final report of the extractable leachable, so to the chemical testing? You know that we have a challenging path to road. And the first, as I said, is prior knowledge. So every information is crucial to start this kind of project. And it leads to the components, the correct component selection that is up to the pharmaceutical industries, of course. And this selection, of course, goes directly to the need or the excludance of the extractable studies. And the extractable studies is strictly linked to the leachable studies or the simulation study when leachable study is not possible. And the last, but not the least part, is the extractable and leachable correlation. This is a fundamental part because extractable profiling has to merge with the leachable part, with the leachable outcome. And this is quite fundamental for two main reasons. The first reason is that with a correlation, we can know where is the problem, where is the real risk, who is the main component that is about concern. And the second part is that if we have a real concern, how can we manage it? And how can we minimize this risk? So it leads to a mitigation step. And finally, we have the final report for the regulatory bodies. But as I said, the two main steps are the first two. So the prior knowledge and the component selection. How can they merge together? They can merge with the risk assessment protocol and documents that here is highlighted with a risk ranking score. So every contact materials that come in contact with the drug formulation during the manufacturing process and or the primary packagings involved can be scored. So we can define if it is in compliance with that specific process and that specific drug formulation or not. And to do that, we can use different factors. The intrinsic processing factors, like the distance along the production stream, and all the chemistry and the chemical factors, so exposure temperature, the duration of the exposure of the drug formulation with the materials, the chemical interaction, and of course also the surface area volume ratio. So all the factors together gives us as an output a score, a ranking of this, of the possible risk. So we can have three main risks, so the low risk, the medium risk, and the high risk. with a low risk, we can react only with a documental evaluation, while, of course, with a medium and high risk, we can move on with an extractable test, and so with the leaching part. And this is the main part. So what about the chemical testing? The study approach between extractable and leachable are quite different. In some points are the same, but it's quite different. As I said in the first part of the presentation, extractable has to be considered as the worst case condition. So it's a laboratory condition. We have to extract all the possible compounds that can become a risk for the patients. While for the leachable, we have to consider the in-use condition because we have to evaluate the reality of the risk. For the evaluation of the compounds, in the extractable part, we have to know what came out because we don't know what we want to see. We don't know what we are searching for. Why for the leachable part, we know what we are searching for because we have an extractable part that depicted as a framework. So the questions from what came out, we have what does came out. And so the aim of the study, of course, is different because for the extractable part, we need an identification of all the contamination compounds. While for the leachables, we need to know how many, so the quantification of these compounds. And finally, the analytical approach, of course, reflects all these differences and all these needs. So for the extractable part, we have to work in a screening approach because, as I said, we don't know what we are searching for. So we are searching for everything. While for the leachable part, we have a targeted analysis because we know that these compounds is about concern. So we have to quantify it and to target it, of course. But in general, for the analytical methodologies, we need to work with orthogonal techniques. So we involve different equipments in order to have a total screening of the molecules. So starting, for example, for an untargeted screening of all non-volatile compounds with LC-Q-TOF, going down to the screening of all volatile compounds with the headspace GC-mass, the screening of the semi-volatile compounds with the liquid GC-mass, and the targeted screening of the metals with the ICP-MS, and the ions with the ion chromatography. But how can we evaluate if these compounds is about concern or not? We need to calculate a threshold. That is the analytical evaluation threshold, and is based on both the sensitivity of the methods, of course, and the safety threshold. So we need a toxicological prior information to calculate the analytical threshold. For example, we need information like the dosage of the drug formulation, the duration of the treatment, and the route of administration. And from this information, we set, let me say, like a limit upon which we have a distinction. So all the molecules that are above the AT has to be identified because can be about concern, while all the molecules that are below this threshold do not have to be identified because they are considered as safety. So we saw that these are the three main points for the extractable and leachable project. So we had like an head for we have to start with risk management. And within risk management, we can perform the extractable testing and the leachable part. And you see that these are merging all together because we have to think about a risk-based approach that consider the risk from the start to the end, so from the early development to the final product. So, in conclusion, what can we take home today from this presentation? Of course, the importance of the risk management and the risk-based approach, because testing scope has to be tailored upon a specific drug product. And we have to consider all the risk factors along the manufacturing process, considering all the components and considering also, of course, the primary packaging. So all the risks of the leaching of every part that come in contact with the drug formulation and finally with the patient. And of course, this kind of project, you can see that are quite complex and time-consuming. So an early planning, it's quite fundamental in this critical part of the risk management. And integrate the extractable leachable strategy into the development of the CMC, it's quite important and fundamental. And finally, I recommend a strong partnership between all the actors that can come in contact with in this project. So from the suppliers of the contact materials and the primary packaging to the analytical expert and toxicological one, of course. So, thank you.